Thanks, Paolo, and thanks, Harm, for organizing and giving us the chance once again to speak at this amazing meeting and to share with you our most recent achievements. This doesn't really work. Okay, thank you. Please note that we are a publicly traded company, so I will make forward-looking statements throughout this presentation. So MolMed, you heard this morning from Janet how the field is evolving in the last 5 to 10 years. Well, we've been around for 22 years, when basically cell and gene therapy wasn't even a field. If you had such a meeting like today, it will probably show up less than 10 companies. But we were there. We were initially set up as a CDMO with kind of 20 people providing gene therapy services for the hospital next door. And we were able to grow up throughout this last 20 years to 200 people, making a solid stream of revenues deriving from our CDMO's activities, 2 facilities serving a lot of different clients across the globe, but not only the hospital next door, and a great network of partners. So our characterizing feature is to have this dual business model, where on one side we keep the initial CDMO business that has been, as I say, grow year on year, double digit for 20 years in a row now. And on the side, we still have our basically main core business now that is developing our proprietary product, especially in the oncology business. So today I will try to cover very quickly both this business that we have and provide you an overview of those. Into presenting you our CDMO activities, I really want to just give you a couple of figures to have that speak by themselves. So in terms of lentiviral and retroviral vector manufacturing, up to date, we have been manufacturing and released over 150 GMP batches. With those, we also manipulated and created the drug product for over 250 patients that were enrolled across over 20 clinical trials. So I hope these figures can really give you the sense of what we are doing and the size and quality at which we are working today. One characterizing feature of what we are trying to serve is to provide so-called one-stop shop solutions. So it can get quite complicated and frustrating for most of the small, medium-sized, but to a certain extent also to large pharma, to manage such a great network of supplier that goes from plasmid manufacturer, then you go to the vector supplier. You may have some internal development around the cell transduction process, but you may also have an external CDMO doing the GMP work. Analytics, most of the cases you want to interact with the external developer of QC methods. And finally, you also have generally some consultancy around QA and regulatory. Well, what we are trying to do here is really providing a single point of contact, managing all different services with just one company. So we, under the plasmid manufacturing, we don't do it ourselves. We, of course, can supply our own plasmid and cell bank, and we can, through a network or subcontractor, manage the manufacturing of high-quality GMP-grade plasmid. We then do ourselves, of course, the lentiviral vector and retroviral vector manufacturing at different scales, from 48 liters using cell factories into 200 liters using the POL-iCell 500 disposable bioreactor. Then we use, of course, this vector into cell transduction, where we do have our proprietary processes for manipulating CD34 cells and T cells, but we can as well work with clients' processes or even mixing those two elements. Analytics, we have over 40 people developing and then routinely performing these over 100 QC tests across purity, identity, and potency assay. And that once again allows you to kind of save time and money throughout this entire process. And finally, we can help you with the filing of the CMC package through an INDO and IMPD. This allows a lot of cost saving and also timelines. That is basically the critical issue. So in most of the case, once you have all different suppliers, a delay in the first step translates in a complexity of managing, of rescheduling, cancellation fee throughout the chain. Whereas here, having everything internally, we can really manage the entire process, optimizing timelines and costs for clients. How does that translate into partnership? You can see here how the span of our collaborators going from non-profit organizations like Boston Children's Hospital and Teleton Institute, medium-sized company like Orchard, Selectis, Rocket, all the way into large companies like GSK. And also looking at the kind of services and clinical stage, we can serve all the way to market with the supply of stream Baylis into phase 1-2 or pre-MA clinical trial, as well as some of them are, of course, still in clinical development. The great asset that we're using for both the CDMO businesses and our proprietary pipeline are the 2 GMP facilities that we have nearby Milan. For a combined area of over 50,000 square feet and over 30 grade BSc suite for vector manufacturing and cell manipulation. Both these facilities are authorized for clinical and commercial purposes. And notably, we have the second facility for which we are still in the process of opening new spaces. The Stream 2 will open up some new areas in 19 and 20 for allowing us a further expansion of services and, of course, getting some new collaboration on the CDMO side. Then I will switch to the other business, that is our proprietary pipeline around the oncology business. As you can see, it's well differentiated. We spent a lot of time in the last 12 months trying to reinforce a little bit our early stage pipeline that was a little bit weak. But as you can tell, we have a late stage asset that's called Zalmoxis. Has been actually approved in Europe 2 years ago. I'm not going to go over too much today. The basic news is that across last year, we got it reimbursed in 2 states, Italy and Germany, with a price around $250,000 to $300,000 per patient. So it's very relevant and very much in line with what we heard about CAR-T. Then there are, we identified different players for the commercial partnering of this asset, and they are currently doing the finalization of market access and commercialization of the asset. Of course, due to some of those issues that were discussed in the initial panel around this gene therapy to be commercialized, the ramp-up of our product is relatively slow, so we are now in discussion with our commercial partner in Europe to try to address this slower than expected ramp-up. Then I will touch base very quickly on our second asset, that is 44V6, an autologous CAR-T therapies that is entering clinical stages in these weeks or month. And then I will use the last few minutes to present you our recent innovation around the early stage autologous and allogenic CAR pipeline. So as I said very quickly, our second asset is the CAR 44V6 that I'm basically not going to go into the clinical details and preclinical details as we did it extensively in the last couple of years. What I would like to mention here today is that by the end of August, we presented the package for moving into clinical trials through a European centralized procedure. So we are now in discussion with the authorities, and hopefully by first quarter of next year, we will be able to treat and enroll the first patient. It's going to be a phase 1-2 dose-escalating clinical trial on AML and myeloma. And as you can see on the right-hand side of the slide, we were lucky enough to get involved all the most important clinical centers in Europe. But I will use the last 5 minutes of this talk to go into this innovation of the last 12 months, basically, where we try to reinforce a bit our early stage pipeline, working both on the autologous side, that is our core platform at the moment, as well as set up a new allogenic platform based on the NK cells. When expanding the autologous platform on top of the 44V6, we decided to add some more targets that are still undisclosed at the moment. But what I can tell you is that we choose, of course, targets that are clinically relevant, so expressed in diseases where there is a high unmet clinical need. We are after some target expressed on both hematological and solid tumor that allow us, as we are doing for the 44V6, to have a very quick proof of concept on a model that we know CAR-T works, like hematological tumors, and then switch as soon as we get some efficacy and safety data on a wider market like solid tumors. Then, of course, they need to have a decent safety profile, although our core technology is around suicide gene, so we probably can manage a little bit better than anybody else a higher risk of related to these CAR-T therapies. We are not after the classical names in terms of target like CD19, CD20, BCMA, but we decided to not be a follower, and so we are looking at some targets that are kind of new in this field. Of course, IP was another important piece when decided this new target, and of course, we discussed and choose those in accordance with our great scientific advisory board that is made by two of the best key opinion leaders in this field, like Malcolm Brenner and Giampiero Dotti, as well as another couple of very relevant hematologists like Miguel Perales and Mohamed Moti in Europe. When choosing the Allogenic platform, what we tried to do were really to get some off-the-shelf therapy that could be expandable to can really treat several patients, even hundreds of patients with a single batch, managing the high risk of allogenic therapies that are graft-versus-host disease as well as durability. Again, the competitive scenario was another important aspect that we decided, and taking into account all these four parameters, we decided that NK were among the best candidate for developing a new allogenic therapy, and that was further confirmed by Carl June at ASCO, saying that NK cells is going to be the next big things, and that's really supporting our decision so far. And this is going to be my second to last slide. What we did in this context were to identify a company that's called Glycosem with a technology very synergic to ours, so that we could leverage on each other IP and technology very well. So we, as I presented to you, are able to generate a lentiviral vector at high titers, and we have this proprietary method of transducing hematopoietic stem cells, whereas what Glycosem brings to the table is a great technology in terms of expansion and differentiation of hematopoietic stem cells into NK cells. And so if you mix those two pieces, you can really obtain a huge number of NK CAR-positive cells that hopefully will trigger a great clinical efficacy. Finally, what is our strategy behind this pipeline is really to deliver new CAR therapies to the clinical proof of concept and then licensing those to bigger players. We really heard in the last couple of panels how big companies are entering this field, and they only have the power really to manage market access of this kind of therapies. And therefore, we really look to licensing and co-development with larger players, and then to, of course, reinvest this income in new pipeline and new asset that can bring value to the patient. Thank you so much.